Hello everybody, we're going to take another look at some of the things we have left with Chapter 2, which is our basic review of chemistry, and then we're going to look specifically at carbon, which in our textbook is Chapter 4. Alright, so we've learned that it's important for a biologist to study chemistry, as chemistry is kind of at the foundation of biology, knowing that it's the molecules of life that actually allow life to be possible and make up our structures. We've learned the makeup of matter is atoms. Atoms make up elements. We learned the particles that make up atoms are protons, which are a positive charge, neutrons, neutral charge, and together both of those are in the nucleus of the atom and make up most of the mass of the atom. And then electrons orbit on energy shells outside of the nucleus and have a negative charge and not much of a mass at all when compared to protons and neutrons. So when you look at the periodic table and you see masses, like the periodic table I gave you, the masses down here are mostly a combination of the protons and the neutrons. Alright, some other things for review: that the proton number is the one up here in the upper corner. So for instance, for sodium, its proton number, also called atomic number, is number 11. There's 11 protons in sodium. Then, if it's a neutral atom, then it would have the same amount of electrons as protons, so the same amount of positives as negatives. So there would also be 11 electrons. And there's also this series of numbers in the upper right-hand corner. If you add those up for sodium, it'll add up to 11. And the thing I like about this periodic table is that they're put into the order of the orbitals, or the energy shells, so you know that sodium has 2 in the first shell, 8 in the second, and 1 in the third. So it makes drawing these really quite easy. So looking at this atom, it has 16 electrons, 16 protons, so that must be, I believe that's magnesium. Oh, no, it's sulfur. There we go. Sulfur. The other one's magnesium. 1, 2. Yep, this one is magnesium. All right, way on this side. So in a neutral atom, then we would have the same amount of protons as electrons. All right. Some other things that we need to know. How to find neutrons. To get the neutrons, you take the mass, such as in carbon, the mass is 12, just round that to the nearest whole number, and then minus the atomic number or minus the number of protons. So 12 minus 6 means that there's 6 neutrons. Keep in mind there are not always the same amount of protons as neutrons. And so what we see here is a collection of the elements. Let's see, 1 through about 90 are found in nature. A bunch of them get cooked up in a lab. And I think we're up to about 115 now, maybe more. This one only goes up to 107. And they haven't even completely named them all, but that's all right. All of these are just an average mass of the atom that you find in nature. So carbon-12, meaning it has the weight of 12, it's atomic mass units. Carbon-12 is just an average, but sometimes carbon comes a little bit heavier. There's carbon-13 and carbon-14 or carbon-11. So carbon-12, this one shown right here, is just the average carbon. So when you have different masses, like say carbon-14, 14 minus 6 would be 8, so that one's heavier because it has extra neutrons. Extra neutrons adds weight to the atom. All right, so remember these are just averages. When you have different masses of the same element, like again, carbon-14, carbon-12, carbon-13, carbon-11, those are all called isotopes. Different number of neutrons, they're heavier. Some isotopes are stable, some are unstable, and then we can use them in different technological applications in biology. Some, though, can be very hazardous. So you've also looked already at drawing atoms, and the two maybe most, well, I don't know if they're the most important bonds, but two of the strongest bonds, the covalent and ionic bonds. Covalent are the strongest bonds, ionic second strongest. Why do they behave that way? What causes atoms to want to bond with other atoms? Because it's this bonding that sets them up to make molecules, which then build all of our biological structures. So you have to remember this, that in order to be happy, atoms like to have a full outer level. The first level, the first shell, can only hold 2 electrons. So if it has 2 out there, like for instance helium has 2 in its first and last level, since it's such a small atom, there it is right there, the 2 out there makes it happy. In the next level, the second shell, it can hold 8. All right, that last shell is called the valence shell. And if there's 8 out there, that makes that atom happy. Or if it's full. As you go out further and further, either 8 or having a full outer level, makes atoms very happy. All right, if you don't have a full outer level or you don't have 8 out there, then you're not happy, and you're either going to want to give up your outer electrons in the valence shell, or you're going to want to steal or share those electrons. So we learned with ionic bonds, like sodium chloride, that sodium tends to give up that one, and chlorine says, hey, I'll take it. And so they fit together very nicely, that one wants to give one away and one wants to take it. So that works very fine. So then that makes an ionic bond. These two charged atoms, now that they have different positive and negative charges, so say sodium has 11 protons, 11 pluses in here, and then only 10 electrons, 10 negatives. So that gives it a net +1 charge. Whereas chlorine, which is number 17 on your periodic table, at least I hope it is, because I don't have one in front of me right now, but number 17 on the periodic table, there's 17 protons, 17 pluses in here, and then it gained an extra one, so now it has 18 negatives, so it gives it a -1, a net negative charge. So opposites attract. These two atoms will want to sit right next to one another because of that charged attraction. So both of these are now called ions. A positive ion is a cation, as it says right there, and a negative is an anion. I remember that by how I love cats. So of course they're positive, right? Cats are the positive ones. That's just how I remember it. So then I want you to look at this, and you might even want to draw this on your own periodic table. When you look over here on this side, these are nonmetals over here. Nonmetals tend to be, on this side, tend to be very electronegative. What does that mean? It means that they tend to want to steal electrons. They have a pretty powerful pull for electrons, or what we call a great affinity for electrons. Fluorine is the most electronegative atom on the periodic table. And then it decreases as you go towards the metals. And the metals are over here. So here's that sodium. Sodium and these guys over here tend to give away or have a less affinity for their electrons in the valence shell. So these are not very electronegative at all. So when you team a very electronegative atom like chlorine with another non-electronegative atom like sodium, chlorine is going to win that electron. All right, keep in mind group number 18, that's the noble gases. If you take a look for all of them, their outer level or valence shell is full or has 8. So group 18 is already happy the way they are. So they're not involved with many chemical reactions. So we don't talk about them much, if at all, in biology. But the ones that form bonds over here with the electronegative and those that want to give up atom or give up electrons, those are ones that we talk about more so. So now I want to have you take a look at this. So there's a cube here. It's a salt cube. The green little balls are chlorine, and they're all negatively charged because they've gained an electron. And the sodiums are yellow. They're all positive 1 charged because they've all given up their valence electron. And so they're all ions. So we basically have a bunch of cations and anions here. And because of that, the opposites of charge, they're not just connected to or attracted to the one that they formed an ionic bond with. They actually have an attraction for everybody else nearby who is oppositely charged to them. So you get this cool structure, like this crystal lattice structure that you see here. Those are called intermolecular bonds. So it's more than just the ionic bond of, let's say these two are connected with their ionic bond, but this chlorine here is also attracted to any other sodium nearby who has that positive 1 charge. Same thing with the sodium, who's ionically bonded to this chlorine. It's also attracted to any of the other green ones around it. So there's all these bonds holding this together, this crystal lattice structure. So collectively, that makes this very strong. All right, very, very strong. But it's also going to make it very able to dissolve in water. So when you put a salt cube in water, it dissolves relatively easily because water also is a charged molecule and has positive and negative ends, which then get attracted to all the positives and negatives in here, and it dissolves this very nicely. So that's one thing about ionic bonds, is that they create ions, which then creates intermolecular bonds where everybody's kind of attracted to their neighbors, not just the one they're married to with their ionic bond. All right, then you learned about covalent bonds. These are our strongest bonds, very important bonds that hold biomolecules together. Those big macromolecules like proteins, nucleic acids, carbohydrates, and lipids. In this case, usually with a covalent bond, when that forms, it's usually a case where the 2 atoms or more than 2 atoms are coming together, and they're kind of from around the similar area of electronegativity, so nobody's quite strong enough to pull the electrons away from the valence shell of the neighbor, so they end up sharing. So there's no stealing here, it's all about sharing. For instance, 2 hydrogen atoms sharing their electrons. So then these electrons, this one belonged to this one, that one belonged to that one, and now these 2 electrons just kind of start orbiting in between them. And so both hydrogens kind of now think that, oh, my outer level is full, so now I'm happy. So they don't go out and seek any other bonds. So this is H2, and we can show this bond, this sharing covalent bond, with a line in between. Again, this is a very strong and very stable bond. And it's good that it's stable because it builds a lot of the molecules that make us. And we can have double bonds. So like when oxygen gets together with another oxygen, they actually share 2 pairs. And we can show that with 2 lines. One that I don't have a picture of is when two nitrogens get together, they actually share electrons in 3 places and make a triple bond. And that is extremely strong. And here's a very common one where we've got carbon surrounded by hydrogen. Now carbon is a very special atom or element because it's kind of in the middle with the 8 rule, where it has 4 electrons in its valence shell. So what it tends to do then is form 4 covalent bonds with something else. So here it's forming those covalent bonds with hydrogen. So here we've made methane, kind of what we call our basic organic molecule. Water is special. It makes covalent bonds with the oxygen to each of the hydrogens. We'll talk a little bit more about water as kind of its own thing, but it turns out that oxygen, when it shares those electrons with hydrogen, is kind of a little more stronger, just a tish more stronger than the hydrogen. So the sharing is a little bit unequal. So those electrons that would be moving around here between the two spend a little more time on the oxygen side than they would on the hydrogen side. Not enough to pull them away or anything, but it makes oxygen have just a little bit of a negative charge. And then the hydrogens just a little bit of a net positive charge. So we say water is a charged molecule or a polar molecule. It has polar ends, a negative end and then a positive end. And because of the way it works out, it kind of bends the molecule and makes it look like Mickey Mouse. So sometimes we nickname water the Mickey Mouse molecule. Now this is going to be important for many reasons. That one of them I already told you, that water then, because it's charged, is good at dissolving other charged things like salts. And that's kind of important because many of the molecules and things in our body are in liquid water inside of us, and it stays dissolved, and we want those things to be dissolved. Luckily, not everything dissolves in water, like fat does not, because fat is nonpolar. So nonpolar things do not like polar things like water. So that's going to lead to many interesting properties. So again, the reason being is that it turns out that oxygen and hydrogen have very similar electronegativities. Oxygen is just not quite strong enough to pull those electrons away from hydrogen. So it just kind of shares them unequally. So here's what water then tends to do. Since water then has polar ends, the positive end of one water molecule will tend to attract to the negative oxygen end of nearby water molecules. That makes what's called a hydrogen bond, which is a type of intermolecular bond because it's in between these two molecules. And collectively, it can make for some strong bonds, not like ionic or covalent bonds, but enough that it makes water kind of sticky and stick together. Hydrogen bonds also make the two sides of our DNA stick together. So it's kind of weak, but it's good that they're there. We want DNA to kind of have a weak bond holding the middle parts together because sometimes we need to separate the two sides of DNA to replicate it or to make proteins. The last type of bond has to do with this guy. They're called van der Waals. And these are pretty weak. This little guy over there. So what it turns out to be is that one surface has all of its atoms. So think about the hand of this little guy here, that he's got all his little atoms with their electrons moving around the nuclei inside his hands, and when he puts his hands up against a surface, that surface also has all these different atoms, again, also with their distribution of electrons and moving around. And so collectively, sometimes you get these areas where you get the negative in his hand in one spot and the positive against the wall. And if it was just one spot, that wouldn't be enough to hold him on the wall. But since he's got all this different surface area material on his hands, all these hairs, that it ends up that he can stick to the wall because of the collective interactions of all these van der Waals. So it's like all of his atoms have kind of a opposites attract with the wall that he's sticking his hands to. And it wouldn't work that way, but he has millions of hairs to increase the surface area. So it makes it pretty strong to hold him up. He can walk up walls. So that's pretty much the weakest of our bonds. So again, covalent is the strongest, ionic second, then hydrogen bonds, and then van der Waals is fourth. One last area, structure determines function, remember? So it's very important with the shape of molecules. So here is a cell membrane, and we have some proteins on the cell membrane. These are receptor proteins that receive certain molecules, but only if they have a certain shape. So these receptors will receive morphine, which is a drug that eases pain. So when we make drugs, or when you make natural endorphins, this little area right here kind of acts just like morphine. And then it gets received by that receptor protein in the right spot, and it causes the correct changes to happen in the cell to ease pain. So it's very important the way molecules are structured, that they work correctly in our bodies. So now you should be able to do all of these, identify the 6 major elements of the body, distinguish between the different parts here of atoms, explain the role of electrons in chemical bonding, then distinguish between the different kinds of bonds, and then know their order of strength. Realize that structure determines function, so shape is very important to the way in which molecules work. And then the fact that atoms bonding together create the different molecules like proteins, carbohydrates, lipids, and nucleic acids that play major roles in our body, the way they function. Now we're going to jump over and look at carbon. When we talk about carbon, we're talking organic chemistry, because there's something so special about it that anything living on Earth, it's based around carbon, or we say it's organic. You might recognize this here. We saw it a little bit ago. Carbon is in the middle, forming 4 bonds with hydrogens around it. So carbon we find in all 4 of these molecules, these macromolecules that build us: carbs, lipids, proteins, and nucleic acids. They play an important role in building them and helping to make them such big molecules. Then there's the carbon cycle. Remember the photosynthesis equation? That plants take CO2, there's our carbon, out of the air, combine with water and sunlight, and then they make sugar that organisms can feed upon. And then those organisms do respiration, which then puts the carbon back into the atmosphere. And that's a huge part of our carbon cycle. Not to even mention the burning of oil and coal and natural gas, which also puts carbon back into the atmosphere. So we say carbon is very special, it's very versatile because of the way it wants to do 4 bonds. So we say it's tetravalent. Tetra means 4. It wants 4 stable, and that's important, stable covalent bonds so that our proteins, our carbohydrates, our nucleic acids, which are DNA and RNA, and our lipids are very, very stable molecules. Despite being very big molecules, they're very stable. So when we build carbon compounds, it's kind of like Tinker Toys. I used to have Tinker Toys. And it looks like this. Our most simple one is CH4, taking carbon and 4 covalent bonds with hydrogen. There's our Tinker Toy model there. But look what we can do with carbon. Instead of putting a hydrogen here, we can put another carbon and make C2H6, which is ethane. Sometimes we can even put a double bond in between them. So our simplest ones, called hydrocarbons, then have just the single bond with the carbon to a hydrogen, like methane over here. These would be nonpolar. They would not be soluble in water, which is good because we don't want everything to dissolve in water. We want some things to hold up. We also say it's hydrophobic. That's going to mean that it does not like water when it's hydrophobic. It's very stable. It's not attracted to the neighbors. Like we saw water, the different atoms in water are attracted to the neighbors, or in sodium chloride, they're attracted to the neighbors. With carbon and hydrogen here, that's not the case. They just want to be with each other. Methane is a gas at room temperature. I'll also say it's flammable. And when you pass gas, there is methane in there. Not that you've ever done that, right? Alright, so then we can start building our Tinker Toys here. So all of the white balls are carbons. The red ones are hydrogens. And we can make long chains of them, or short. We can branch one way. We can make ring structures like this one over here. So look how neat carbon is and all the different things that we can do with it. And these are just small. We can make long, huge chains of them. It's a very diverse molecule. There's just another view of the chains and the rings. Isomers are when you have the same molecular formula, but it forms a different structure. Now 2 of these here are isomers. Can you tell which 2? That would be these 2 here. Both of these have 6 carbons, so C6, and they each have 14 hydrogens. So they're both C6H14, but they have different structures. One is a long chain, the other has a branch. But they're isomers because of the same molecular formula. So I don't know if you know this, but both glucose and fructose are isomers of each other. They're both C6H12O6. So there's another example of isomers. There's some isomers there. These are called geometric isomers. They kind of crisscross, but would have the same molecular formula. These are interesting. These are isomers that are stereo images of one another, and so they're called enantiomers. They're mirror images, I guess you could say. If you look here, the thumb is pointing this way, the NH2 is over here, but on this one the NH2 is over there. So there's literally like a left-handed and a right-handed version of both of these molecules. So those are called enantiomers. We have to be careful with enantiomers. Remember, structure determines function, and so receptor molecules may be set up only to receive one of the enantiomers because of the shape has to be just right and oriented just right. So for instance with certain medicines. There could be an L version, a left version, and then one that they call a D version. One of them will work, but the other one won't. Like with a pain reliever, one will work, the other won't. This is kind of scary. With methamphetamines, one is a decongestant sold over the counter, but its enantiomer is actually the highly addictive stimulant crank. Got to watch out for those enantiomers. This one's kind of scary. This is called thalidomide. There's an S thalidomide, it's the left one, and the right one is the R thalidomide. They used to give this to pregnant women in the 50s to help them with their morning sickness. But look what it did. It caused babies to be malformed with their limbs kind of just budding. Very scary. Of course, they pulled that off the market very quickly. If any of you are music lovers and like Billy Joel, his song "We Didn't Start the Fire" has the children of thalidomide in it. You'll have to listen for that next time. And something that we always want to watch out for when making drugs and we don't quite know all of the effects on them. Alright, so look, we can do some more things with carbon molecules. So this is ethane. It has its typical carbons with hydrogens. But let's take this hydrogen off here and let's put an O and an H instead, which is what we call either a hydroxyl group or an alcohol group. So now we've made it ethanol. So both of these, though we didn't change them very much, have very different properties. One's a gas, one's a liquid. One's polar, one's nonpolar, charged versus non-charged. And the biological effects of them are very different. So this little end here with the OH, that's called a functional group. It allows us to change a molecule's properties without changing its molecular formula very much. In this case, we just took off an H and added an OH. So that's our next job, is to learn the functional groups and be able to recognize them. Here's another example with hormones, estrogen and testosterone. Notice how they have a very similar structure. So when carbon forms rings, we just draw them like this, and we think at each point then there's a carbon there, and we don't even bother drawing the hydrogens or the other atoms that are in there. But we can see where the functional groups are. There's that OH. CH3 is another functional group. So they look very similar. Testosterone and estrogen, not very different molecules, but look at the different effects that they have just because of these different functional groups. So what we like to call viva la différence, right? So here's the functional groups that you will need to know. They're in red. The hydroxyl group, carbonyl, carboxyl, methyl, amino, sulfhydryl, and phosphate. I want you to make flashcards of these with also their molecular formula and their drawing so that you can recognize them. And you'll need to know these for the whole rest of the year and be able to recognize them. And you don't have to memorize all this stuff. I just want you to be able to recognize the functional group. So the OH we've already done. Here's what carbonyl looks like. C with an H and a double bond to an O. Carboxyl looks a lot like carbonyl. You got to watch out. Sometimes you think, oh, that's a carbonyl group, but look more closely. Look for the C to the double bond O and then an OH down here. The amino, very important one in proteins and amino acids, the NH2. Sulfhydryl, the SH, important one in proteins. Phosphate, PO4, very, very important in both nucleic acids and the phospholipids that make up our cell membranes. Oh, and I should also say ATP has phosphate groups. The methyl group is a CH3. That becomes important when we talk about methylation of DNA. And they are our building blocks for biological molecules. All right, so go ahead and make those flashcards for those functional groups, and you should know those by heart for the whole rest of the year. All right, there they are again. There's 7 of them. All right, let me know again if you have any questions.